much. I'm sure. I'm not sure what was said, but I'm sure it was very nice. Thank you for the introduction. So I hope you can all hear me. I'm wearing this mic, but there we go. So I have to apologize for hijacking the whole meeting. I didn't realize that I had an option to not do everything that was thrown my way. So I've basically said yes to everything, and now I've basically, you're going to hear a lot from me. So hopefully you'll find it interesting. So I'm going to start today by talking about local modeling of the diffusion signal. I gave a talk yesterday, it was more about tractography. So let's proceed. So here's what I'm going to talk about. So I'm going to give a brief introduction to what I mean by microstructure modeling and what's been done. Then approaches that I have worked on for modeling single shell data and then modeling multi-shell data, and then conclude from that. So starting with the microstructural imaging. So microstructural characterization or imaging has been the kind of unique selling point for MRI, for diffusion MRI for decades. This is really what it was introduced to look at. It's been the subject of intense research over the years, and we've seen a lot of very impressive work in this area. But despite all of this, it's remarkable how few methods actually deliver on that promise of giving us microstructural information. So one of the issues is that a lot of the models that are used assume a single fiber population, and that's the real problem because when we look in the brain, there's a lot more than one single population in any given voxel. We have crossing fibers and fiber dispersion, and all of this makes a big difference to the inferences that are made. And a lot of these models also require really complicated, complex, and custom acquisition strategies that are impractical for routine use. And I'm much more interested in routine use than these blue sky kind of things. However, I do think that there's some really useful information that we can get quite easily. And if we focus on the very dominant effect, which is just partial volume, how much stuff is there? I think that's already a very interesting piece of information, which is a kind of first order effect. Anything else that you then look at is a kind of secondary modulation of that main term. And I'll explain what I mean by all of this. So anyway, what are we looking at really? So white matter is what I tend to focus on, but it's now diffusion MRI is being used for other types of tissue, but primarily white matter is the one that people have been looking at. And most of the models are going to be looking at this kind of vision or perspective on how white matter looks like. So you've got a dense set of cylinders, basically, which is what these axons look like as far as diffusion is concerned. If we look at the individual axons, this is a peripheral nerve, but it's kind of very similar in the brain. It wouldn't be a Schwann cell, it would be an oligodendrocyte in the brain. But the structure is very much the same. We've got the axon membrane, and within that we've got some structure. And around the axon, we have these myelin sheaths, which are basically the cell membrane of the oligodendrocyte in the brain, wrapping around the nucleus. And the purpose of that is to provide faster conduction speeds and insulation to the electrical signals. If we look inside the interaxonal compartment, there's also a lot of structure there. So we've got the myelin, which wraps around the axon. We've also inside got microtubules and neurofilaments, all of which may play a part in the diffusion signal to some extent. So if we look at all the factors that might influence the diffusion signal, this is not a comprehensive list, but it's pretty much everything I can think of like this. It's reviewed very well, and Christian Beaulieu's got a very good review of this stuff. But there's a whole lot, there's massive amounts of different aspects of the microstructure that might influence the signal. And if we look through this list and look at what really makes a big difference, we know axonal membranes are a massive driver for the anisotropy that we measure in vivo. We know the density of the axons is crucial because it gives rise to more of that, more signal from that. One that is really important, and has been the focus of a lot of my work over the years, is this arrangement of the fiber bundles. So how crossing fibers, curving fibers, diverging fibers, all kinds of different arrangements that you can think of which affect the signal. Partial volume effects with other tissue types. So I think of crossing fibers as a partial volume effect between two different fiber bundles that happen to have a different orientation. For me, it's kind of a similar problem. but when you have other tissue types as well, they have different characteristics in their signals, so it becomes more complicated again. Other things that really get very little attention, but are really quite important, myelination. A lot of the models completely ignore the myelin, and I think that's a really big problem, and we're starting to see some work in this area. The intrinsic diffusion coefficient of water within each compartment, typically we assume literature values, but of course that's going to make a difference to what we think we're going to see. We have all sorts of other cells present in white matter. It's not just axons. We have oligodendrocytes, which are the things that myelinate the axons, astroglia, and when we have inflammation, you're going to have all sorts of other cell types coming in as well. So all of these things are very rarely included in any model. I think there's only one I can think of that has looked at cell bodies as such, as a compartment. People are starting to look at blood flow as something that might be a confound rather than anything else. So this is to do with intravoxel and coherent motion. Initially, diffusion was proposed as a method of looking at perfusion through this effect at the very low b-value range. So it's a confound and something we need to bear in mind, but not necessarily get too worried about, I think. Stuff that doesn't make much difference. The other one is T2 in various compartments. That's completely ignored at the moment. The fact that CSF has a very different T2 from the intraaxonal compartment, or maybe the extraaxonal compartment might have a different T2 as well. All that's kind of ignored, and I'll explain why that's a problem a bit later. But recently, there's been a lot of work on axonal radius and the distribution of axonal radii and how much difference that makes, and whether we can measure this distribution of axonal radii. And then recent work has kind of shown that it's actually extremely difficult to get any kind of contrast from that. I'll talk about that a bit as well. Exchange between the two compartments. Most models completely ignore exchange, and I think they're probably right to ignore it. But it is, again, something that you might want to worry about. And that's just basically how fast does one molecule of water move from one compartment to the other over the timescale of your experiment. And if there's a lot of exchange between them, and of course you're going to see kind of averaging effect, so it changes the assumptions. I'll talk about that again. So Christian Boulay did a lot of work looking at the effect of these kinds of things, organelles and transport mechanisms. They don't really play a part in diffusion, so we can ignore them. So basically this is a kind of like my list of what matters and what we still need to get a handle on. Okay, so that's just kind of setting the scene. Exchange is something I mentioned, so that's just about how fast does water exchange between, for example, the intraaxonal compartment and the extraaxonal compartment. If we can assume slow exchange, so over the timescale of the experiment there's basically not a lot of exchange happening, then we can assume that the signals from the two compartments are adding up linearly. So if you assume a single exponential model, the bit from the intraaxonal compartment adds linearly with the bit from the extraaxonal compartment, and you're fine. But if you've got fast exchange, so that's basically molecules move from one to the other and back constantly, then what you get is basically the intra and extraaxonal compartments basically give you one averaged diffusion coefficient, and you have no hope of then disentangling the intra from extra, at least not very easily. So that's why it matters. So that's exchange looking at intra to extracellular environments, but the other way I think about exchange is much more on a macro kind of scale. If you have this kind of configuration of fibers, then you can treat diffusion within that whole thing as kind of coherent. Gives you one type of signal for the whole lot, even if there is exchange locally between the intra and extracellular environments. So if the bundle size is much larger than the displacement of the water molecules, then you can still treat that whole thing, that whole bulk bit of tissue, as not exchanging with the other one. And the reason that kind of makes a difference is because you can then measure the signal for that whole bundle and treat it as one unit, if you want. That's your fundamental unit of microstructure. And in that case, the signal from that bit adds up with the signal from that bit linearly. The only kind of scope for any exchange is at the interface between the two bundles, which is a very, very small fraction of the total volume, so you can ignore it. But if you have this kind of interdigitation of things, then water molecules will probe both environments over the timescale of the experiment, and then you do have basically a violation of that slow exchange property. So then in that case, we may not be able to assume that the signals between the two compartments add up. But that all assumes, of course, that there is a significant amount of exchange from inter to extra before that even becomes a problem. But most people completely ignore that. And I think if you look at microstructure histology slides, you find that actually this is overwhelmingly how the brain organizes itself. It's very rare to find that kind of really axonal level interdigitation, although it might happen. So that's just about the exchange assumption. I think we can assume slow exchange. The other thing that's not talked about a great deal is the effect of the pulse duration. When we do a pulse gradient spin echo experiment, you apply a gradient in one direction, put a 180 and apply the same gradient again. A lot of the theory that underpins microstructure imaging is based on the assumption that the pulse is narrow. And what that means is that you tag the position of the spin instantaneously before it's had a chance to really move, let it move, and then untag it instantaneously again. So that all relies on the pulse being so narrow in time, so short, that the displacement of the water molecule is negligible over the timescale of that pulse. The problem is in practice, even on the best scanners, even in preclinical scanners, we can't do that. This, if that's an axon, that's about a micron or less. The root mean square displacement of water, in vivo is much, much greater than a micron. So even with the shortest pulses, we're going to get, this is basically the path of, say, that molecule over the timescale of that pulse. And what that does is then the interpretation of the Q-space signal, which I haven't gone into, but I'll go in a second. When you look at displacement, then we're talking about displacement between the average position during the first pulse to the average displacement between the second pulse. Okay. Now, what that means is that when you have a restricted environment and a long pulse duration, then the average position of your spin over the timescale of your pulse will self-average to basically the center of the compartment. Okay, so if it's long enough, it'll just basically on average be right in the center. And the same for the second on-tag pulse, and in which case you're looking at the difference, the displacement between an average spin in the middle and an average spin in the middle, so you get zero net displacement with a long diffusion pulse in a restricted environment, which means you get no attenuation. And that's interesting because in an axonal environment, which is restricted, the transverse signal really doesn't decay very much at all. And you can verify that in experiments as a function of the diffusion pulse duration. With the spin duration, the pulse durations that are available to us, we basically don't see a great deal of attenuation in the transverse plane for the intra-axonal compartment only. And that really plays in our favor for imaging of microstructure, at least the fiber structure, because that means we actually get more signal than we thought, which is nice, I think. But what it does mean is that for axonal diameters, we don't have sensitivity to axonal diameters. So if you do a really simple experiment and you look at the signal that you get in a restricted cylinder in the transverse plane as a function of the radius of that cylinder, or diameter, sorry, for kind of clinically realistic pulse durations, then we really get no difference in the signal all the way up to about 6 microns. And it's only then that you start to see a difference in the signal that we measure. And that's a bit of a problem because the biologically relevant range is well below 6 microns. So this is corpus callosum. There are much larger axons than 6 micron in the brain. Some of them go up to 10, 20 microns, but they're quite rare. And the overwhelming majority are 1 micron or less. So that's kind of been borne out recently by some work from Ivana Drobnak from the UCL lab. So this is a lab that's been doing a lot of work on axon diameter measurements, and they've just come out to really say that in practice, we're not actually sensitive to axonal diameters. Which is a bit of a problem because there's been a lot of studies done looking at axonal diameters and showing differences in various pathologies, and you've got to start wondering what it was that was making a difference to this signal. And that's a theme that I find kind of comes off quite a lot. The models that we have are quite simple, and there's a lot of ways to affect the signal that aren't included in the model. So when you see a difference and you interpret it as one thing, there's a very good chance that it's actually something else. We just don't know. But For us, the fact that we're not sensitive to axon diameters is probably a good thing, because we rely on this assumption for NODI, for example, and apparent fiber density. Basically, we're saying the signal that we measure there for that intra-axonal compartment, it's just about how much water there is in that intra-axonal compartment transverse to the fibers. It's not about how many of these are large axons or not. It's just how much is intra-axonal. So that's quite a nice way of interpreting the signal. So a lot of microstructure modeling is done is assuming the Q-space framework, so spin propagator framework. So just to kind of recap briefly what that is, if you have a sample with fiber orientations lining up in this kind of orientation, then the displacement probability function, which is basically how likely it is that a molecule starting at some random position within the sample moves by a certain amount or a certain distance in a certain direction, you get this kind of probability distribution function. So it's quite likely to diffuse along that direction, quite likely to diffuse along that direction. So that kind of gives you a full characterization of the diffusion process in that sample. And it's great because it turns out that there's a Fourier relationship between the signal as a function of Q, which is basically a measure of the gradient and its direction. So it's this kind of 3D space of the gradient vector between Q and the spin propagator as a function of displacement. So this is great. What we have to do is measure the signal as a function of Q over the kind of 3D Q space on top of the 3D spatial domain, and then we can get the spin propagator at every voxel. So there's a few problems with that. One is that it relies on this narrow pulse approximation that I talked about earlier. So it relies on the fact that you're tagging instantaneously and on-tagging instantaneously, and it doesn't happen in practice. But it's not as problematic as it seems because of what I spoke about recently. It's just a different interpretation to what you're seeing. My bigger issue is what does this propagator give you in terms of microstructural information? It's a nice characterization of the diffusion process, but what you really want is some kind of biological interpretation to what you're seeing. You want to know about, the fiber density, the myelin levels, whatever, something that is actually biologically interpretable. And you can't really get that from a spin propagator because it's so model-free. It's just what it is. You need a model to then extract some information from it. So from my point of view, if we want microstructure information, you're better off modeling it right from the beginning and then fitting to the signal directly, rather than going via the spin propagator framework. So that's just my take on that side of things. So just to recap on most white matter models so far in common use, they will assume multiple non-exchanging compartments. So that's the slow exchange approximation I was talking about. And the intra-axonal compartment will be restricted diffusion in cylinders, a bit like this, if you kind of think about spin within that, or there for that little one. And in the extra-external compartment is a hindered diffusion model. So there's no restriction as to how far they can go, but they are hindered compared to what their normal diffusion coefficient might be. And those assumptions are basically common to a whole load of methods. Out of these, probably spherical deconvolution is the one that makes the least assumptions. I think this is the only one we do make. But all of them are essentially the same set of assumptions. Dispersion is something that is not talked about a great deal, but the NODI model is based on the fact that it models dispersion. Most models assume that axons are perfectly straight and perfectly parallel, and it just never happens. The brain is not that simple. Even this is corpus callosum. Look at the directions there. This is the one bit that we assume has got a nice single fiber population. It doesn't. These massive angles in there. And there's some other bit in this is actually kind of thalamus. So it's not unexpected to find so much dispersion there, but you can see that it's pretty far from a single fiber orientation.
Excuse me. Is that cartography performed on?
No. This is tracers, or EM.
Okay.
This paper. This is the Max Planck Institute. They do some amazing stuff with EM, serial EM sectioning and imaging. But it's well worth having a look at that. It's slightly frightening when you see these things. And the problem is that has profound influences on the estimated parameters, because if you're just a little bit off axis, your model of your signal perpendicular, you're going to get a massive, especially at the high B values, much more attenuation than you'd expect if you were straightly, white perpendicular. So if you've got a mixture of different angles, then, and you're assuming they're all straight, it's not going to fit very well. In fact, it's going to fit pretty badly. So that's a big confound that is rarely accounted for. So how do I go about modeling diffusion data? So I started off modeling single shell data, and the main reason for that is because it's easy to get. You can still get that in a clinic in a feasible amount of time, and we use it pretty routinely, so it can be done. So the first thing to look at is the crossing fiber problem, which is by far the biggest problem that we have here. So if you have single fibers here, that's the signal that you'd expect to see at a constant B value over the sphere. It's nice and easy. Nice big signal tangential across the fibers and big loss of signal along the fibers. And that obviously tracks with the orientation of the fiber bundles. If you have a mixture of those two, then what you have is a kind of average of these two things, and that's what it looks like. So it's a funny shape, but you can see it's got structure in there that we can use to then recover the fiber orientations. And the way we do that is by first measuring enough signal for that. So to be able to capture the kind of information that we have in the signal, we do need to have a lot more orientations sampling, much denser sampling than we would for tensor imaging. So that's led to this whole Hardy acquisition methodology, where we sample the signal at constant B value over the sphere. So a large number of directions, I'd say at least 50 or so, but 45 is kind of lower limit. It depends what B value you go to. The optimal number of directions depends on the B value. I've got a paper on that if you really want to look into it, but basically you want to be able to capture these little details in the diffusion signal, in the orientation structure. So just to look at that B value dependence in more detail, obviously if you have a B value 0, you just got a sphere. It doesn't matter what orientation you look at, you're going to get the same signal. As you increase the B value, you get, that's B value 1000 for a crossing, just simple model, a tensor model. You see, start to get this structure when you cut through this plane. When you increase the B value, and that angular dependence becomes more pronounced, so you get more contrast. But you also get a loss in signal. Now this is assuming a tensor model, so in fact that decay is exaggerated compared to what you actually see in actual real data because of the restricted thing that I was talking about earlier. But you get the point that there is a compromise between how much signal loss you've got and how much contrast you've imposed. So typically I would recommend something between 2,000 and 3,000 as a good B value there, because you get kind of pretty good contrast to noise in the angular domain there, even though the images don't look very good individually. So to put it all together, the spherical deconvolution framework is the way that I kind of put it, but it's essentially a generalization of a multi-tensor, if you want, fit or something, where you just say I've got different orientations together, and obviously if I know the orientations present and I know what each orientation, what the signal would be for that orientation, then you can predict what the signal should be. And that can be cast as a spherical convolution operation. So if we measure that and we have a good estimate of what this looks like, and we assume that it is the same across all fiber populations, which is one of the big assumptions in spherical deconvolution, then we can recover the fiber orientation distribution function, or fiber orientation density function, fiber orientation distribution. There's lots of words for it, but I call it fiber orientation distribution. So how do we do this? So this being a mathematical meeting, I thought I'd put in a bit of maths just to keep everybody happy. But we use a spherical harmonic framework, and there's a lot of reasons for this, but it's got very nice properties, and it's a very concise representation for functions on the sphere. It's got very strong parallels with the Fourier series in Cartesian space, and in particular, it has the equivalent of the convolution theorem in the spherical harmonic basis. Now in diffusion imaging, we can make a bunch of assumptions about the symmetry of the problem. One is that the diffusion process is symmetric. The probability of a molecule moving from A to B is the same as the probability of a molecule going from B to A. Anything else is flow. In this case, we don't have any odd terms in this series. All the odd terms are 0, because the odd terms correspond to antisymmetric functions. Also, we know that they're real. The signal's real. The diffusion signal is real. So that also means that we can modify the spherical harmonic basis to look at just the real components of what would otherwise be a complex series. So this is what they look like for different harmonic orders. And you got this phase term, which gives, you get more phases at different harmonic orders. So we can represent the diffusion signal as a linear combination of these basis functions. We can represent the fiber orientation distribution as a linear combination of these basis functions. So the process of spherical convolution, we'll kind of look at it as an analogy with the Cartesian case. In Cartesian case, we got the Fourier transform of A convolved with B is a product of the Fourier transforms of the individual functions. In the spherical case, it's a little bit more complicated, and it took me a while to get my head around it. Maybe some of you will find it quite intuitive, but I'm sure most won't. In the Cartesian case, you have to think as convolution as the action of a distribution of translations on a distribution of positions. So although that suggests that basically the A and B are equivalent in terms of how they're treated in the convolution, really it's much easier to think of that as being basically distribution of translations. So maybe that's kind of a blurring operator on positions, which is your image or something like that. And in this case, you can start thinking about the convolution on a sphere as the action of a distribution of rotations on a distribution of orientations. So then gets really messy because rotations you can't represent as spherical harmonics because there's three parameters for a rotation, the three Euler angles, whereas the spherical coordinates are on just two coordinates. And you have to represent them using this thing called rotational harmonics, which is a kind of, I guess, the equivalent basis to spherical harmonics, but for rotations. And what that gives you is a full m by m matrix for each harmonic order. So before we had a vector, an m-sized vector per harmonic order. Now we've got an m by m matrix per harmonic order. But when that happens, the spherical convolution is actually quite simple. For each harmonic order, the spherical harmonic transform of A convolved with B is the product of the matrix, which is a rotational harmonic transform of the first one, by the vector, which is a spherical harmonic transform of the second one. So it's a matrix vector operation. It's linear, it's nice and simple per harmonic order. But now the functions A and B are not equivalent. You've got to represent one as rotational harmonics and the other as spherical harmonics. So conceptually, they're actually quite different entities. But we're in luck because in diffusion imaging, or at least in spherical deconvolution as it applies to diffusion, the problem is really simplified because the response function is axially symmetric. And what that means, if in practice, it's quite hard to derive it in a way that actually makes sense, and I don't think I've really managed to do it satisfactorily. But if, say, A, for example, we assume now is a response function, if it's actually symmetric, then all diagonal terms are zero. You can actually go through the series and find out that it is actually zero. But it's better than that, again, because the power in this is a fiber orientation distribution, and that's the signal. The power is going to be invariant under rotations. No matter which way you spin the fiber orientation distribution, the signal will spin along with it. It's going to be the same power. And that means that basically your rotational harmonic transform reduces to a single scalar. Okay, so that says it's diagonal, and that says all elements on a diagonal have to be the same. Otherwise, you can't keep the power. So now all you have is one term per harmonic order to represent your response function. So it's very nice and very fast to do. So how do we do spherical deconvolution with that? Well, it's just a straight linear mean squares fit. You've got you're trying to minimize the mean square error between your measurements. Hang on, I've got things here. So that's the coefficients of the fiber orientation distribution. Then you convolve your current estimate with the response functions. So that's just multiplying by a diagonal matrix, essentially, if you concatenate all the harmonic orders. Then you map these SH coefficients to amplitudes along your gradient directions. And then you take the difference between your predicted and actual measurements and minimize the error between that. That's very simple. But if you just do that, that's what you get. It's not very nice. It's a deconvolution. They're always very sensitive to noise, and you get a bit of trouble. So you can do something about that by low-pass filtering, because most of the noise is in the high angular frequency terms, because those terms are quite low amplitude in the response function, so they get amplified. So you low-pass filter, you get something that gives you something that's kind of more representative of the two fiber populations that I know I've put in there. But actually, we can do a lot better than that by constraining the negative orientations. So we know this is a fiber density function. It's supposed to be positive definite, or at least semi-definite. So if we constrain these negative orientations to be 0 or close to 0, we do a lot better, and that gives us a huge gain in noise suppression. And the reason why it works is because this function is generally pretty sparse. So it's mostly zeros along most of the domain, and you've only got a few kind of peaks coming out of the noise. So it does a really good job of killing the noise. So how do we do that? The way it's done currently is that we add this regularization term to the previous equation, which basically maps the SH coefficients of the fiber orientation distribution to those amplitudes where the distribution is negative. And then we had a strength of, it's a regularizer, so that controls the strength of that regularization. And then we minimize that whole lot. The problem here is that this matrix here will depend on the fiber orientation distribution, because that just maps those bits that are negative. So we iterate, and in practice we do that very, very quickly. So within 5 iterations you've converged, and it's relatively stable. And there's a couple of other bits and pieces that you can do to regularize that a bit further and make it more stable. But basically that's how it works. And it does a pretty good job. So this is tensor in this region of the brain, and this is spherical deconvolution, giving you really radically different information to what you had before. What does this fiber orientation distribution really tell us? So this comes back to some of the points I made earlier. If we look at a bit we have white matter fibers, the signal that we get in this part of the corpus callosum, this is corpus callosum signal at B3000, you get exactly what you'd expect to see, so very strong signal in the radial plane and pretty much nothing in the axial plane. And that's, when you do this spherical deconvolution, gives you an FOD that has a peak in the right direction. And the great thing is, because it's a linear operation, you know that the signal scales with the amplitude of this thing. So the greater the signal in the radial plane here, the larger the amplitude of the fiber orientation distribution. Yes?
Sorry, what is the max number of directions that you can deconvolve in?
It's a distribution. There's no set maximum. You just get the distribution. So as long as you've got sufficient resolution, so your series goes up to a higher enough angular, you don't truncate too early, then you should be able to recover quite a lot of directions. In practice, there's the signal itself is kind of band limited, so you're kind of stuck at harmonic orders around 6 or 8 in practice, and that would put a limit on the number of directions that you can get. I don't like to talk about number of directions because I think we rarely have single directions in practice, even in corpus callosum. It looks like a single direction, but that's because the way I've done the deconvolution, I've already kind of included, if you want, the dispersion that is inherent to a white matter bundle. But actually, there's dispersion, there's curving, there's all sorts of stuff. So I prefer to keep things as a distribution, which gives you much more of a feel for what is actually going on, and then trying to map it to a direction explicitly. So if we look in the kind of cut through of this cartoon of white matter here, so this is basically the kind of classic way of looking at white matter in all of these microstructure models. What you can imagine is happening is, if you have a sufficiently high B value, then the extra axonal signal is essentially attenuated so strongly that its contribution is negligible. It's very small. The myelin signal you don't see in diffusion imaging generally, because it's a short T2 component, and we have to use quite long T2s in diffusion because of all the massive gradients that we have to put on, especially at high B values. So that means that we've basically left with just the intra-axonal signal, which we showed earlier with large pulses that we use in the clinic, is essentially proportional to the interaxonal volume and not modulated by the axon diameters. So it's a really nice, clean interpretation. Now, this is just kind of a cartoon of the white matter, and of course things are more complicated than that. But I think this is a really useful way of thinking about the signal that we get from spherical deconvolution and how to interpret it. It's not perfect, but I think it's pretty good. So just to recap some of the key concepts behind spherical deconvolution, which are more or less implicit in what I've said, but I'll just pull them out now. So it's a multi-compartment mixture model, explicitly. If you kind of take set directions and you say I've got a large number of fixed directions each with their own signals and volume fractions, and I add the map. If you let that number of directions tend to infinity, you end up with spherical deconvolution. It's exactly the same thing. And here the compartments consist of differently oriented sections of white matter, and it's assuming a slow exchange again. The big assumption is that all white matter bundles look the same. So the kind of half-full interpretation of that is that there's no variation in microstructure. There's no point doing microstructure imaging because we have no contrast there. The way I like to see it is basically what I'm saying is variations in microstructure have a negligible effect on the signal. It might have a little effect, but it's really hard to pick out when you've got this background of enormous differences in dispersion, crossing fibers, all this other stuff that makes a big difference. So I'm not saying that there is no effect of microstructure on the signal at that level, but I think it's very small compared to the other stuff. The other thing is that we assume that there's no extra-axonal compartment, and that's because we rely on these high B values to get rid of them. In practice, a lot of people use spherical deconvolution with lower B values, like B1000 or something, where the extra-axonal compartment is still there. That's not such a big deal as long as the response function that you measure includes that compartment within it. So now we're not looking at the signal as kind of a compartment, which is intra-axonal and an extra-axonal compartment. I'm just saying I have a response that represents what a fiber bundle looks like, including all its other bits and pieces that go with the extra-axonal compartment, the cell bodies, everything is included in that response function. And then we just assume that that is relatively invariant across the brain. And the other big thing here is just what I was just kind of talking about. There is no explicit microstructure model here. We've put everything about the microstructure in the response. Extra-axonal, intra-axonal, everything's in there. So it's a nice way of factoring out all the microstructure stuff and focusing on the orientations and the volume fractions of these orientations. But it's not great at getting the kinds of microstructure models that people have been trying to get. And the final point here, which is quite a technical detail, but I think might interest some people in the audience, hopefully, is the issue of normalization of this fiber orientation density function. In a matrix, we don't do any normalization of the signal whatsoever. And the idea is to retain that proportionality between the fiber density that we get and the diffusion signal. And I'll explain what I mean by that in a second. So this is a very complicated slide, and I was hoping it was going to animate itself, but I've obviously failed. But anyway, let's go through it one step at a time. So pretty much all the other methods that I've seen in the literature work on the signal attenuation. So that means the signal normalized to the B0 signal. So you take S over S0. And the rationale being that that makes it more quantitative in some way. I tend to disagree with that. I think this is only true if the B0 signal is pretty much the same across all the different compartments that might be present in your tissue, and I don't think that's true. So if we look at the B0 signal, the B0 signal here, for a voxel containing some CSF and some white matter, you have the volume fraction of CSF times the signal that you get for the CSF at B0, plus the volume fraction of white matter times the signal that you get for white matter at B0. And the same thing at B3000. If you then go and normalize it, you have to bear in mind that the signal for CSF is about 3 times what it is for white matter, on average. So it's very bright because it's got a longer T2 and a slightly higher proton density anyway. The signal for the CSF at B3000 is essentially 0. It's gone. We've crushed it. So if you put it all together, then the signal at B3000 over the signal at B0 for this case, you do the math and you get this kind of equation here. And the point here is that is not proportional anymore to the volume fraction of the white matter, which is what you would have liked it to be. If I kind of give you a kind of brief example of that, here's the equation I had earlier. If you had pure white matter, then the ratio is fine. That's what it is. If you have a voxel with half white matter and half CSF, then you've got a signal that's basically a quarter of what that was. Okay? If you go through the maths, it's kind of falls out quite easily. The point is here, volume fraction 1, volume fraction a half, and yet my estimate of my fiber orientation density estimate from that will be a quarter of what it was. And it's all about the fact that you've got very different sensitivities to different compartments because of their different T2s. It makes a massive difference. So I'm not saying that not normalizing is the answer, but certainly I think normalizing to the B0 signal has problems that need to be really thought about. It's not just, it doesn't make it more quantitative in my mind. I mean, that's a massive problem. And it means the signal is not proportional to volume fraction and the operation is not linear anymore. So that's why I don't like it. The other thing that some people like to do is to normalize the fiber orientation distribution to have unit integral. And that's based on the premise that probably the name of it is sometimes called the fiber orientation density function, orientation density function. It sounds like probability density function, so therefore it should have unit integral. I don't agree with that. I don't think it is a probability density function. I think it is a density function, but not a probability density function. And it's a problem when you have, again, exactly the same thing. Partial volume. Consider voxel, half white matter, half CSF. DW signal is dominated by the white matter signal. Okay? And that signal, when you have half white matter, is approximately half what it was for the pure white matter voxel, right? You have half the signal that you had for the pure white matter voxel case at the high B value. And so if you normalize your FOD but to have a unit integral, then your predicted signal from that is going to be twice as high as the measured signal because you scaled it up. So if you enforce that normalization to happen as part of the optimization, you're going to have a poor fit. You're going to have massive residuals here just because you've imposed that unit normalization to the FOD. And that causes other problems. One is, again, you have an FOD amplitude that's no longer representative of volume fraction. I think that's a problem. I like to have an interpretation. If you think of it as probability, that's fine. That's what you want. But if you want it to be, the apparent fiber density, basically, you don't want to be normalizing it at all. And also, it means that your noise distribution is not uniform. And that's the same for the normalization to the B0 as well. Less of an issue, but still something to worry about. Okay, so avoiding normalizations, if you operate purely in the raw signals, you preserve the linearity of the system, your apparent fiber density is proportional to the signal, but you have to carefully calibrate your response to scale up with the diffusion signal. If you've got a different scanner and it spits out data that has twice the amplitude, then your response has to be twice as large to compensate for that. And you also need to worry about bias fields and effects like that. When you have strong variations in the signal intensity, you have to correct for that because otherwise, if you have a single response, it's going to not work very well. So there's a few issues to get right before you can do quantitative things with that approach. So how do we get this response function? So there's different approaches possible. Most people will assume a model. So they'll just say, I've got a tensor model. I'll specify the FA as being 0.8 or something, and the mean diffusivity as being 800 or something, 900. And then I'll get an estimate for what the signal looks like from a diffusion tensor model. I prefer to measure it from the data for the reasons that I mentioned before. We don't actually know what the microstructure is. We don't know that the tensor model is a good fit. We know the tensor model doesn't work as a function of B values very well. It's actually it predicts much faster decay than you actually get in practice. So why not just measure it from the data? So what we do is we perform a first pass, which is a low order CSD with a rough estimate of what the response should look like, so you know roughly what it's going to look like. Identify voxels that have single fibers in them, and that's basically based on the peak amplitude ratio between the primary peak and the secondary peak. And then you average the response, you average the signal from the top 300 voxels that you've identified by just reorienting their signals and averaging that. And then you do a CSD again on using that response, and repeat from here again until you get convergence in your response function. And with that, you get very quickly convergence to something that looks a bit like this, which is quite representative of what the signal should be for a single fiber population. So note that response is going to include, because it's estimated from a voxel, from a whole voxel or a whole set of whole voxels, it's going to include any effects that are inherent to a white matter tract, like the fact that there's other cell types present, there's a bit of extracellular signal, maybe that there is dispersion inherently in any white matter tract. All of this gets kind of bundled into this thing. So it's quite robust from that point of view to other effects. Okay, so that's basically how spherical deconvolution works. And after the break, I'll be talking about what we can do with these kinds of things in terms of group analyses and group comparisons. The other thing that I'm not talking about is tractography using these kinds of things, because I already talked about that yesterday, and I don't want to bore the same people again. So multi-shell data offers opportunities.
Excuse me. When you say that you detect the voxel with a single fiber, and then you relate the signals, it means that you relate the signals and the vectors may not coincide with the other vectors.
No, so the way it's done in practice is actually using spherical convolution. It's quite neat from that point of view. I fit the spherical harmonic series to the signal, and then I say I can do that with a delta function aligned along some other orientation, and that reorients it.
Parametric model.
Yeah, basically. And then you sum all of that series.
In the new orientation. That's okay.
You sum all of that across your 300 voxels, and then you discard all the non-axially symmetric terms. It's essentially how it's done now, and it's very fast that way. But the way it used to be done was more about taking the gradient vectors, reorienting them, then doing the spherical harmonic fit to just the axially symmetric terms and summing up. And there's different ways of framing the same thing, but it's implementation rather than concept. But yeah. So for multi-shell data, we have great opportunities. So the initial motivation-ish for this, even though that's not how I saw it, but spherical deconvolution inherently, as it was framed, was unable to really exploit the multi-shell information. In fact, you could easily extend spherical deconvolution to take account of the multiple shells, and it's very easy to do that, but it doesn't really help in terms of resolving crossing fibers, because actually the signal from the low shells is so much greater, so much higher amplitude than the high shells, that it dominates the fit. And then you get a loss of angular resolution because those terms have reduced, they're more band limited at the lower B values than the high B values. So it was actually counterproductive to use multi-shell data in CSD as it was. But there was something we could do. One of the problems that we had with CSD is contamination from non-white matter tissue, so gray matter and CSF. CSF is a problem because even when you crush it out, you get magnitude data coming out, so the noise is rectified, there's a bias, so you're going to get non-zero outputs. If we could have zero mean data, I'm pretty sure this would get close to zero if you could correct for the Ricin bias, but we can't very well, so you end up with a lot of noisy stuff in the ventricles, and on noisy data sets, that really is a problem for tractography and other analyses. We also get really messy stuff on the gray matter. So what can we do about this? And that also is a problem when you do tractography, because it's not uncommon, for example, to track from one gyrus to the next in ways that are clearly not physical. So we came up with this idea of multi-tissue CSD. So the idea is to extend spherical deconvolution to multi-shell. That's easy to do, but now allow multiple ODFs from distinct tissue types simultaneously. So it's a multi-compartment model, and it relies on the idea that different tissue types would have a distinct B-value dependence. Okay, so their decay, rate of decay as a function of B-value will be sufficiently different that you can actually resolve them when you have multi-shell data. And again, all of these things are measured from the data themselves. So the way to do this is let's estimate what the tissue responses are. So it's exactly the same process as for what I just spoke about, but now we're looking at 3 different tissue types. In this case, CSF, very simple. Let's measure the isotropic signal from the ventricles, and that's what it looks like as a function of B value. There's no orientation dependence to that, so we just model it as a single scalar. So whilst you could say it has an ODF, really the ODF is just isotropic, so it's just a scalar. It's just a density value. Same for grey matter. You kind of pick out grey matter regions that are, as best you can tell, pure grey matter, and that's the signal you get from that, the green curve here. So it starts off higher than white matter and goes lower than white matter. And again, the white matter stuff, you identify single fiber voxels from the white matter, and that's the isotropic part of that signal. But now the white matter has a full-blown response per B value. Okay, so you've got a much more rich kind of response for that than you had before. These other ones that were isotropic, if you think about it, all it is basically a sphere that gets smaller as you go. But it's essentially what it is. So with that, we have an estimate of what each tissue type, what its canonical response function is. So we can plug everything into exactly the same framework as we had before. This maths is identical. We've got now a vector of parameters. That includes the full spherical harmonic coefficients for the FOD and the other tissue components as well, which are just scalars because it's isotropic. And that gets mapped to signals, and these signals now have multiple B values in them, so you have enough information to tease them all apart. And you then add this constraint that all of these densities, so for a full-blown ODF, you're then mapping to amplitudes along on the sphere. But all of these densities have to be positive semi-definite. So you can't have negative white matter, negative CSF or whatever coming out. In this case, we've had to use a kind of more hard constraint, quadratic programming type constraints, just because otherwise a soft regularizer just doesn't really do the job very well. It becomes very dependent on exactly the strength of the regularization. So we went for a hard constraint on this one, which is unfortunate because it's a bit slower to run. But anyway, you can work it out. And it does an incredible job on your data. So this is the diffusion data displayed as the estimated densities of gray white matter CSF and color-coded as such. These are not, it's not a classification. These are the actual, not, I wouldn't call them grayscale, but they're densities. They're not classified. This is a T1 segmentation that corresponds to the same patient in this data. So this decomposition has no spatial prior imposed on it. It is purely done on the voxel-wise signal. And it does a really good job of segmenting out gray, white matter, and CSF purely from the signal. And even the deep gray matter structures come out as a bit of a mix between gray and white matter, which is exactly what they are. So it works really well from that point of view. And here's some other kind of views of the same data, but if we focus on this part of the ventricle here, this is what we get with single tissue, single shell, single tissue CSD, which is the previous generation. Now what happens is we've got completely rid of the CSF part, and that's because all the CSF signals have been attributed to the CSF component, and the white matter component is free to be as clean as it wants. So we've completely cleaned up the ventricles, and in the grey matter part, same thing. Note that we still get these radial projections here in the cortex, but by and large the grey matter signal has been moved over to its grey matter component, and the white matter component remains. So this is nice to see these kinds of structures because we know there's going to be axons projecting quite deep into grey matter, but not necessarily uniformly and not necessarily with a very strong density. And the effect on tractography is also quite impressive. So if you look at the whole brain tractography using current single shell technique, it does a good job, but it's quite messy at the boundaries, and you get a lot of some streamlines kind of joining between gyri. But if we now track on the FOD, white matter FOD from the multi-tissue CSD, you get a much, much cleaner tractography. So it doesn't project through that way. And that's just because we now have fiber orientations that, we're not allowing the algorithm to track noisy FODs anymore because they're not there. So that does a really good job. So the multi-tissue CSD provides volume fractions, basically directly from the data. It's fast and it's intuitive, and it's minimal assumptions. So what we assume is that we've got distinct compartments in slow exchange, just like every other model does, and that the signature of these tissue types is invariant. So it's the same, gray matter is the same no matter where, and white matter is the same no matter where, and the same for CSF. So the current implementation, we consider gray matter, white matter, CSF only, but the important thing here is the framework is not limited to just that. And it's going to show you what I mean by that. In a minute, sorry, I'll talk about something else first. So there's been some work recently trying to estimate these tissue responses in a much more unsupervised way. Currently what we do is we take a T1 image, segment it into grey, white matter, CSF, resample it to the diffusion-weighted images, and then pick out voxels that have very pure tissues. But we can also use methods that look at non-negative matrix factorizations as a way of getting these the voxels from which to measure the responses. And the basic idea is that the data is positive definite. It's a signal, it's a diffusion signal. And it's a linear combination of positive definite responses. So the inherent signal per tissue type is going to be positive definite, weighted by positive tissue densities, the densities. So everything is positive in this thing. So we have this set up. So the signal is the product of the responses by the weights. And all of these matrices are positive definite. So we can apply these non-negative matrix factorization approaches. They're a little bit unstable, but they do a kind of relatively decent job of getting somewhere. If you just do it as is, you get this kind of decomposition. It's not very clean, and you get the not very nice looking decay curves. What you can do is add constraints like a sparsity constraint, and then you get much more believable results from that. You can also then go and iterate by then looking at voxels that have very strong densities in this map, and then measuring the response from that and doing it again. And then this is what you get from the anatomical segmentation, just to compare the two outputs. So this advantage here is this is completely data-driven, no automatic, no intervention from the user. And more recently, Dan Christians has come out with a much more sophisticated framework, which is called convex non-negative spherical factorization, which I have to admit I'm a bit hazy on because it seems really complicated. But it's essentially a non-negative matrix factorization where you now assume that the responses are linear positive combinations of the actual per voxel signals, which is pretty much what we assumed anyway. And we want to maximize the separation between the responses within this domain. And what it does is it pushes the kind of responses out to the outer edges of the domain, and so you have a convex hull. It's all very complicated. I'm not sure I'm explaining it very well, but if you look at the paper, it's a Mikai paper, you might make more sense of it than I did initially. But he's framed it, and basically it's a blind deconvolution problem where you're trying to estimate both the responses and the fiber orientation distributions such that basically everything is positive, not just that, and you have this approach there. Whatever, convexity is about the way the problem is framed rather than the way it's optimized. I took a while to kind of get my head around that one. And the way it actually works in practice is it alternates between estimating the responses and alternating and the fiber orientation distributions, the ODFs, and alternates between them. But that does a really good job of it. So this is the three densities for white matter, grey matter, CSF, fully automatic, unsupervised, and that's the white matter ODF in different data sets. So I think this is HCP and this is a more regular data set. But where it gets really exciting is when you try and apply this to pathology. So far we've just looked at grey matter, white matter, CSF. Here's a tumor patient with had a resection here, and there's edema. And if you do this unsupervised response function estimation and you tell it I want 4 responses, you get a 4th component, which is the edema component, coming out very cleanly. And that overlaps with the white matter part. So you can see this is the white matter ODFs superimposed on the edema density. And it corresponds quite well with the T2 image as well. So this is really promising stuff to start looking at microstructure from a purely data-driven point of view and get information that really looks biologically important. So this is very much the kind of thing that we're looking into at the moment, specifically for neonatal data, where we really don't have much of a clue what we're looking at. There's not the microstructure models don't really apply to neonates. And I don't know if I've got time to carry on for a bit.
I don't know, it's up to you. Maybe
I've only got a couple of slides. I'm just going to go through them. So far we talked about tissue types, but actually the framework is much more general than that, because you can put microstructural compartments in there if you want to. As long as they add up linearly, it doesn't matter whether you talk about tissue types or tissue compartments. And so what that means is, for example, the great matter CSF decomposition that we have here, we could cast it again as a different combination of microstructural compartments. So it could be that the gray matter response that we've estimated, because we've estimated it from a particular voxel or set of voxels, is actually, you could look at it as being some amount of intra-axonal plus some amount of extra-axonal plus some amount of CSF signal, and the white matter response would be some different contributions from these different things, and so on and so forth. But the problem now is how to derive responses. So the way it's currently done is very simple to deal with, because we're measuring the signal from distinct locations where we assume we have a pure tissue type. If we now want to get intra-axonal, or compartment-specific responses, there's no way to do that, because no single voxel is going to be a single compartment. So that makes it very tricky, but we might be able to apply microstructure models to do that kind of work. But this is something that we really want to push for the neonates as well, because of the issues. It's a very different brain. I don't know if I want to talk about that, but I'm not going to talk about that. It's probably slightly esoteric, but anyway. The other thing is the framework is not limited to diffusion imaging or variations in B value. You can add shells with different diffusion times, echo time, gradient patterns, whatever you can think of. You can put oscillating gradients in there, all kinds of patterns, and you could put different modalities even if you want to in that framework. It's all additional kind of shells if you want, or additional data. And the problem is always to derive these appropriate responses. So to conclude, microstructure imaging is, I think, incredibly hard. I think the models that have been using are really looking for a needle in the haystack, because models where you have a parametric representation of the microstructure and then you want to infer these parameters, there's too many variables. It's a degenerate problem. Different people model the microstructure in different ways from the same data, and they get effects that are very good fits to the data, but it's a completely different interpretation of the data. It's a very degenerate problem. So the approach we've taken is much more empirically driven. We just want to know, different tissue types have different signals. If we can measure these inherent responses, then we can do the decomposition, and that's irrespective of the underlying microstructure. All we want to know is, regardless of what the microstructure actually is for that particular compartment or tissue type, what's the signal that we'd get for that? That's the only consideration. And the main assumption here is that the dominant effect in the data is these differences in volume fractions between these different tissue types or different compartments, or whichever way you've chosen to decompose your data. And I think that's pretty much all I had to say. So yeah, that's it. Thank you very much.